Welcome to a Taconic webinar entitled The Pirc Rat Model of Human Colon Cancer, from the epigenetics of tumor initiation to the study of human gender disparities. Our speaker today is Dr. James Amos Landgraf. Allow me to introduce myself. My name is Megan McBride, and I will be your moderator today. Before we get started, I'd like to address a few commonly asked questions. The presentation will be available at the end of today's session. The link to the presentation will be emailed to all attendees at the end of the week. If time allows, we will be taking questions at the end of the presentation. If during or at the end of the presentation, please type your question in the Q&A panel. Attendees will not be able to see your question, as it will be sent directly to the presenter and will be answered at the end of the session. If you cannot see the control panel, please hover your mouse over the WebEx toolbar at the top of the screen and then select Q&A panel. If you are experiencing any technical difficulties, raise your hand in the control panel and our staff will assist you. We hope you enjoyed today's webinar entitled The Pirc Rat Model of Human Colon Cancer, from the epigenetics of tumor initiation to the study of human gender disparities, presented by Dr. James Amos Landgraf. His graduate work in epigenetics and complex traits led to his current postdoctoral work with William Dove at the University of Wisconsin-Madison. After working with the Murine mouse and realizing the biological and genetic limitations in the mouse, he collaborated with MicroCT in the McArdle Laboratory for Cancer Research to develop the Pirc Rat model of human colon cancer. He was an American Cancer Society Fellow seeking to identify markers of tumor progression using the Pirc Rat model of colon cancer using a CGH expression array and assays of epigenetic silencing. Dr. Amos Landgraf has a continued interest in epigenetics related to cancer development and is exploring the recently recognized gender disparity in human colon cancer. Jim, I turn it over to you.
Great, thank you very much, Megan. I hope everyone can hear me. I'm going to give a generalized background of the Pirc rat, first how we identified the animal, and give you a brief background on colorectal cancer. I'll tell you more about how we're using the Pirc rat in longitudinal modeling of cancer, and talk more about the epigenetics and genetics of tumor development and what we see in some of the crosses that we're doing with the Pirc rat. And then I will touch on the role of hormones in colon cancer development, and this is work that is a collaboration between myself and Gijs van den Brink in the Netherlands, and so I will talk about some of our for publication. So colorectal cancer is the third leading cause of cancer and cancer mortality for both men and women. And many of you that have logged on, if you have interest in colon cancer, you probably already know that. There are many risk factors for colon cancer: age, obesity, especially the Western diet, very high-fat diet, will increase the risk of colon cancer. But recently it's been identified that gender is involved in the initiation and age of onset of colorectal cancer. There are now several large epidemiological studies showing that, and I'll talk more about that in the end, but it's also involved in the severity of the disease. Now, what do we know about colon cancer? There's a lot of work that's been done in the last 20 years, but about 25 years ago here at the university, Amy Moser identified in a mouse, a B6 mouse, mutations in the adenomatous polyposis coli gene. And this was also identified in human mapping studies going on at Johns Hopkins at the time. And they found that mutations in the APC gene, especially those in families that carry familial adenomatous polyposis, but even in 80% of sporadic colorectal cancer, have mutations in the APC gene. And here you can see an image of a colonoscopy of FAP patients where you can see multiple adenomas. And I don't know if you can see my pointer, but you see multiple adenomas developing in the intestine. And this was work done by Pirc here at the university. So there are lots of murine models of colorectal cancer. There are many mouse models of the human disease, including use of somatic chemical mutagens, AOM and FIP, oxymethane. These develop multiple adenomas, often really just single adenomas, and the latency is very long. There are lots of APC mutants. The Murine mouse, of course, which was identified here at the UW. There's a 1638N, which is an insertional mutant in APC, and a deletion, delta 716, that's also mutation in APC, but there are other models of activating mutations that also get colorectal cancer. The rat models have been somewhat limited. Somatic chemical mutagenesis has been used, AOM and FIP, and other models have been used, other chemical mutagens to develop intestinal adenomas. But there's really been a limited use in the past of rat genetic mutants. But I will talk about how we really capitalized on some of the early ENU work done in the mouse to take advantage of that in the rat. So there are many limitations to all of these. There really is no perfect model. The mouse models, however, seem to have interesting differences between the human disease in that their tumor distribution is largely in the small intestine. So min mouse will get anywhere from 60 to 100, even more, 120 tumors in the small intestine, with very limited numbers in the colon. They have a very early morbidity, 90 to 120 days, which is good in the sense that you can do experiments somewhat quickly, but bad if you wanted to follow any kind of progression of any one tumor. The short latency of that time period really limits the amount of time that you can allow the tumor to grow. The size of the mouse is also limiting. So if you want to do any assays of toxicology, the amount of serum that you can get from a mouse is very limited. The rat is probably 10 times the amount of blood, and the amount of tissue and the tumor sizes are much larger in the rat as opposed to the mouse. And this, I can't emphasize this enough, that there are many genetic modifiers in the mouse genome that affect the MIN phenotype at least, and a lot of the other phenotypes as well. And that being said, so if you have other genetic mutations, say in other genes that you wanted to test their effect on the combination of APC mutations and these other phenotypes, if that knockout or knock-in is on a different genetic background, it will dramatically affect your phenotype of the MIN phenotype. Bringing in backgrounds such as 129 can cause dramatic disparities in tumor numbers, so you really can't compare animals unless all of your work is done on congenics in the B6 line itself. And even within the B6 line, we see some potentially spontaneous mutants that are altering the tumor phenotype in the B6 line itself. So on the flip side, in the somatic chemical mutagenesis, the dosage variation, how effective those dosages are in the rat, as well as the long latency, up to a year in rats before you even see a tumor, and less than 100% incidence. So that ranges anywhere from 50 to 90%. So it's very difficult if you have a treated and untreated group to determine if the effect is due to either the preventive effect, the chemopreventive effect, or the treatment of whatever you're treating, or whether it was just a possible dosage variation and less than 100% incidence in those animals. And then on top of that, you get a very heterogeneous mutation spectrum. This is not a controlled environment, so AOM is not something that's present in the environment. So the mutations that arise in these tumors, and there's some question as to whether or not they're indicative of the mutations that you see, although there are activating mutations in beta-catenin that are seen, which is part of the Wnt pathway. It's probably not the environmental type of causative agent that you would see in nature. So to try and control for many of these limitations in the mouse and the chemical mutagenesis models, we've developed this Pirc rat model. And I'll talk about how we developed it, and I'll tell you about the phenotype. So this involved germline mutagenesis. So we treated male Fisher 344 animals from Taconic with ENU. The spermatogonial stem cells were then mutagenized because of this ENU treatment, so all of their offspring were then screened for mutations. We didn't use a phenotype screening. We actually used a targeted gene approach where we PCR amplified a portion of APC from exon 15. This was introduced into yeast along with this universal gap repair plasmid, which would produce a fusion APC add2 protein. So if there were any truncation mutations that were induced in APC by the ENU or frameshift mutations that would cause non-working add2 gene, we'd see red colonies as opposed to the white colonies that would be present. And in fact, here are the yeast plates from the the two sibling rats, 5021 and 5022, and you can see this plate is mostly white, with the red colonies represented in possible errors in the PCR reaction. But in this plate, hopefully you can see that half of the colonies are red and half white, this animal being heterozygous for a newly induced point mutation, and sequencing confirmed that in fact we do have a point mutation. So where does this reside? So if we look at the APC gene, this is a representation of the human gene with all of the components of the different binding domains. These are the two most common FAP mutations, and the hotspot of somatic mutations occurs in this region. Here's the min mutation with the corresponding human codon, the delta 716 deletion mutant, and 1638N. And Pirc happens to lie right within this FAP and hotspot mutation region. So we wanted to know what the phenotype of this was. When we looked at the first collection of Fisher 344 animals that came out of it, the first thing that we noticed was that compared to min mice, where in the small intestine we see a large number of adenomas, anywhere with an average of about 80, in the Pirc rats, half of these animals had no small intestinal tumors, and the number was very small for the remaining ones. When we now shifted and looked at the colon, where really almost half of the min mice that we examined at the time didn't have any colonic tumors, and half did, all of the Pirc rats had colonic tumors in excess of 3, and this was at about, I think we were doing this at about 120 days. So really what we saw was the shift in small intestine, where there's fewer in the Pirc rats, to the colon. We actually don't know why this is occurring and why there's this physiological difference in the mouse, but between the mouse and the rat. This is how it got its name, polyposis in the rat colon, or Pirc. So now I want to tell you a little bit how we're taking advantage of that colonic phenotype to do longitudinal modeling for treatment with chemopreventives, and as well as some more detailed mechanistic models of how tumors develop. So what you're seeing here is a video of colonoscopy. This was colonoscopy was really initiated and developed in Maine, Germany by Neurath, and we're using the Storrs colonoscope to now look at tumor development in these rats. So this is a colonoscopy just as a human would have. You can see tumors developing. So you see these small adenomas and now these very large adenomas. So this is a 90-day-old Fisher animal. And you can see that these are some very large adenomas. This adenoma we would estimate to be over ½ centimeter. As you can see, it occludes nearly a third of the intestine. Now the advantage of using this is now we can document where those tumors arise, and we can come back a month later. So this is now a month later. These two tumors in the proximal region are a bit larger. We can actually see, well, here we see peristalsis occurring, but we can actually see tumors that are now being initiated. Let me see, did this? Oh, so it didn't advance, but here's the next one. So here, now we see that these are larger. And we see these smaller tumors now being initiated here along the length of the intestine. So new tumors seem to arise in the Pirc animals over time. Here's a new cluster of tumors that we didn't see at the previous time point. These tumors grew larger, and now that very large tumor will come out, has now almost occluded half. So these tumors continue to grow through the life of the animal, and this provides an opportunity now to document where these tumors are in each place in the intestine. But now what was interesting is we came back now a month later, so now this is now 90, 120, this is 160 days or 150 days later. You can see a lot of these tumors now have reduced in size. So not only, this is actually the very large tumor that was occluding the intestine before, and you can see that over time, this is a very powerful tool to look at not only growth, but regression of tumors. And so what this allows us to do is document each tumor as opposed to looking at whole animals. So if you did a conventional study where you treated animals with a certain compound and you sacrifice that animal, say 140 or 150 days later, you would see only this time point. You wouldn't really know how many tumors were present in this animal prior to that without colonoscopy. So this is very powerful in that you can use many fewer animals, and instead of documenting the number of animals that you're following and an average tumor number, you're now documenting individual tumors and their responsive rate. And so we did a preliminary study, actually a study that was published, much of this data was published in this Archives of Biochemical and Biophysics on supplementation with vitamin D. And you can see that through documenting with longitudinal colonoscopy, we can see that there are different tumor fates and very different tumor behaviors. In fact, even in the control animals, we see some regressing tumors, a number of static tumors and growing tumors. But you can see, depending on the treatment, we can, and in fact, the aspirin plus vitamin D3, we see that there was a tumor that was regressing. But more importantly, in this NC analog, which we wouldn't know previously with the different treatments, but we saw that a number of these were actually static, even more than in the control compared to some of the other treatments. So it allows you to follow tumors over time, and we think that's a very powerful approach to addressing chemopreventives and chemotherapeutics. The other advantage to doing colonoscopy and doing an in vivo approach is we can take samples of those tumors. So here you're seeing a biopsy of an adenoma. This is a pinch biopsy where we actually can take about a 3 millimeter cubed fragment of the adenoma. We can actually even biopsy the normal epithelium as well. We can get up to 4 micrograms of RNA from that, as well as DNA. We're using the QIAGEN RNA DNA kit, not to plug any one. I know that there are a number of companies that have different kits to isolate both, but it allows us to follow both DNA and RNA. And we can actually even go in and remove the whole tumor by polypectomy. What you're seeing here, sorry about this image isn't as good, but this is a wire loop. We've now looped this wire around this adenoma. We're actually going to pull it, and then actually we can cauterize and actually remove that tumor and actually burn it off the surface, and then we can go back in and actually pick it up and retrieve it with the biopsy forceps. And as we pull out, you can actually see where we actually remove the tumor there. There were other adenomas next to it, but this allows us to retrieve that material and actually take it out of the rat and look at it histologically, as well as taking RNA and DNA. So other ways, one of the issues is that how do we know we're looking at the same tumor? If a tumor goes away, how do we know that was ever there, or where should we be looking? So one of the things that we took a cue from the human is that when human gastroenterologists identify tumors in a human and they want them removed, you can actually go in with this India ink dye and a syringe and actually mark with a tattoo. Here you can see we marked right next to this tumor with this India ink. What that gives you the ability to do is really pinpoint where a tumor is, or if you remove that tumor, where it was, and you can follow that now for, let me advance here. And that tattoo will last actually for the life of the animal. We've looked up to 6 months out from the time that we did it, and there's really no difference from day one. So those tumors will last a long time, and it'll allow us to register these tumors if we want to come back and look. But to be honest, we don't use this technique very often, because really by location and depth, and you can even follow some of the vasculature and some of these very trademark hallmark branching, you can actually come back and really pretty accurately pinpoint where those tumors were. So what can we do with these biopsies? As I said, we can take RNA and DNA. We've done some longitudinal expression arrays using EB arrays from Christina Kondrarsky, which is available for the R coding program. We can identify genes that change over time. And in fact, if we did these longitudinal biopsies, one of the interesting genes that has come up is DCC, which is downregulated in growing tumors. So this is synonymous with what goes on in the human, and that DCC is often deleted in colon cancers that show more advancement. So we think that it's a hallmark of progression in these tumors, but that remains under study. So some of the other techniques that are being used in the mouse that we are also using in the rat are MicroCT and PET. This is a combination MicroCT PET image of using fluorodeoxyglucose, or FDG, to identify Pirc tumors, and we can see that Pirc tumors definitely are metabolizing higher than the surrounding tissue, as indicated by FDG, but we can actually see that there may be some tumors, whether or not this is a fecal pellet or a tumor. We can actually go in by colonoscopy and confirm that, but we see this differential labeling of FDG. So now I want to switch gears a little bit and talk a little bit more about the molecular nature of how these tumors develop, and we found some very interesting results that have some similarity to what we see in the mouse, but we feel that there are some advantages in the rat. So you can imagine that tumors can initiate through many different mechanisms. So again, we're starting with a predisposed tissue in that they're carrying a wild-type copy of APC and the Pirc allele of APC. So all these animals are heterozygous. The homozygotes are inviable. But we know that in tumors, there's loss of heterozygosity where the wild-type allele is lost through some mechanism. And there could be any number of different mechanisms that could result in this. And so we really wanted to pursue understanding the mechanism of loss in these adenomas. So to do that, we developed this pyrosequencing assay for loss of heterozygosity. So not to go into detail as to how this assay works, anyone can Google pyrosequencing, but this really allows you a quantitative measurement of the different alleles within a sample, either DNA or RNA. And we use that to identify loss of heterozygosity in the colon adenomas. And this is all in the in our paper that we published in PNAS. So when we look at tumor development, here we see a normal epithelium centers around 50% contribution of wild type compared to Pirc. This is a non-Pirc carrier, so you can see that it's all Fisher. And in the colon tumors, we see that the majority of the tumors lose heterozygosity. But there is still a substantial number that seem to maintain heterozygosity and look very similar to wild type. And this it does not fit Knutson's two-hit hypothesis. And so we investigated those further. We wanted to know if loss of heterozygosity was occurring through large chromosomal rearrangements. This is common in advanced colorectal cancer, but it's not necessarily common in any of the mouse models of colon cancer. And we wanted to know if this was occurring in the rat. And in fact, it is not. So these tumors seem to develop in the absence of chromosomal instability. So there are no large losses or gain. What you're seeing here are each color represents a different chromosome, and any deviation from the 0 point, either these would be gains or losses in the tumor compared to spleen. This is a control spleen versus Pirc spleen, and we don't see any effect of the Pirc mutation itself on chromosomal instability. But in the tumors, we don't see any instability as well. And these were very large, 1 centimeter tumors coming out of a Pirc rat. So we feel like they can develop and even progress to a carcinoma in situ with invasion into the stalk without any noticeable chromosomal instability. So through the pyrosequencing assay and the array CGH, we've determined that majority of the tumors arise through somatic recombination, whereby there's no loss in copy number, but we do see the Pirc mutation now homozygous and the loss of APC. However, we still had that substantial percentage of tumors that seem to maintain heterozygosity, and it was unknown to us what method it was undergoing to achieve the tumor growth and initiation. So could it be an extragenic mutation, say in activating beta-catenin, or could it be that there was just a second hit in the wild-type copy of APC? And this was probably the one that I thought would be occurring. But the possibility could be that we actually just have transcriptional silencing of APC. So we pursued this a little bit more. We wanted to know, first of all, if loss of heterozygosity was something that was just increased. So maybe this somatic recombination was increased throughout the genome, maybe due to the Pirc mutation. We know that mutations in APC are involved in binding to axin and some of the other components of the spindle. And so we were wondering if that may affect crossing over or somatic recombination in these animals. But we actually, in fact, did not see that. So when we looked at tumors, we did find a single tumor that seemed to lose heterozygosity on chromosome 1, which has some interest in that there are many imprinted genes on the rat chromosome 1. But when we looked across at other chromosomes, especially chromosomes that had other tumor suppressors or tumor activating oncogenes on them. We didn't see any kind of loss of heterozygosity or instability. And in fact, even on the Q arm of chromosome 18, we did not see any loss of heterozygosity. So these were a conundrum. So how could these tumors be developing in the absence of additional mutations? So we really wanted to address this by sequencing. So we sequenced the APC gene in tumors that maintained heterozygosity. We actually found very few mutations. So we only found 3 mutations out of all of the tumors sequenced. And 2 of them were stop codon mutations in exon 15, I guess, as we would expect. But we didn't find any in beta-catenin or KRAS, or we did some sequencing of P53 as well. We did not see any mutations in any of these other genes that may be implicated in adenoma formation. So we wanted to ask the question whether or not epigenetic silencing was playing a role. Here we had a very high percentage of these tumors that seemed to not have a mutation in other genes. Now, certainly there are other mutations in the genome that could very well have been mutated that we just weren't following. We did a very targeted approach and not a whole genome approach. But we did want to rule out the fact that epigenetic silencing was occurring. And in fact, we actually have evidence for epigenetic silencing. So now, this is now a plot of pyrosequencing of cDNA and the corresponding DNA of the same sample. And what you see is that when you see loss of heterozygosity in a tumor, now tumors are all in black and normal epithelium is the open characters here. And we've also treated, and I didn't introduce this, but we have done treatments with the inflammatory agent DSS, which will increase tumor number, but doesn't seem to increase the ratio of whether these tumors maintain heterozygosity or lose heterozygosity. And in this case, all these tumors down here seem to have lost heterozygosity both in their DNA and in their cDNA. You can see that they kind of cluster together. And there were certainly a collection of tumors that looked very much like normal epithelium, where the tumor maintained heterozygosity both in the DNA and in cDNA. But this was the curious class. So these tumors now maintain heterozygosity in DNA. So they have 2 copies, both the wild type and the mutant. But only the mutant allele is expressed, and that it's biased. You would expect if there were equal transcription from both alleles, those tumors would be represented up here. But that's in fact not what we saw. We see transcription of only the mutant allele, suggesting that there is some sort of silencing of the wild type allele that is selected for in these tumors. And in fact, one of the mutations that we found was in an intron 13 of APC. And what that mutation created was an alternative splice form. So you can see this smaller fragment here that is specific for this alternative transcript, which creates a premature stop codon. And because it is not in the last exon, and because it's near the splice junction, this is likely subjected to nonsense-mediated decay. All right, so our summary so far of the mechanism, and I've just really brushed upon some of these points, is that tumors, we can follow them longitudinally via optical colonoscopy. We can follow some longitudinal expression changes using RNA that we isolated. These tumors seem to develop with a stable genome. The majority of adenomas lose wild-type APC through somatic recombination. And then tumors can develop via alternative pathways. And in fact, a certain percentage maintain heterozygosity. So we're actually in the process of pursuing the mechanism of tumor formation, especially in these epigenetic silencing. So approximately 30% of the tumors maintain heterozygosity. This is in the ACI by Fisher F1. I didn't emphasize that, but in these F1s, approximately 30% of the tumors maintain heterozygosity. It's slightly lower in the straight Fisher line. But then approximately 10% of all tumors seem to be undergoing this epigenetic silencing event. So now I want to switch gears again and talk briefly about the genetics. I briefly mentioned that we're using an ACI by Fisher F1 to do some of these experiments where they're now informative across the genome at all heterozygous locations. So I'm going to tell you our experiences with some of the crosses that we've done, and then talk, just briefly touch on some of the congenics that we've created. So in the Fisher line, this is maybe a slightly underrepresented male Fisher animal where we see tumors in the colon. This is the proximal colon and the distal colon. And some of these actually didn't show up very well on the image here, but there are some smaller adenomas that develop down here. They average about 8 colonic tumors at about 6 months of age. When we look at the F1, these tumors are much larger. They seem to appear earlier. Unfortunately, these animals do become moribund earlier than Fisher, so if you want to follow anything for a much longer time, I would not suggest using at least this particular F1. But when we continue to backcross this animal, you can see that this looks very much like the human FAP condition, where there are just a multitude, really a carpet of adenomas that develop in the intestine. Now, as we've now created in excess of N10 generation on ACI, this tumor number has actually decreased, and it looks more similar to maybe the N2, somewhere between the N2 and the F1. So that tumor number seems to relax. It's not as severe as this in the N3. So we believe that there are both dominant and recessive enhancers, and that there's a lot of interaction and epistatic interaction of multiple genes in the genome that affect this phenotype. So as a summary, so here on the Fisher 344 at 7 to 13 months of age, they get somewhere between 7 and 14. You can see that the number is dramatically different between the males and the females. If you treat these Pirc animals with ENU and you look at 7 months of age, we can increase that number dramatically. And presumably ENU is a point mutagen, so now we're affecting that second allele and making that initiation process much quicker. And so they can develop up to 100 tumors in their colon by 7 months of age. We've looked at the different crosses in Wistefirth, a different strain of Fisher that we received from Japan, Brown Norway, which is the sequence strain in the rat, and ACI. And you can see that in the F1, Brown Norway looks very similar to some of the other strains, in fact, maybe even a little enhancement, but as we created a congenic of Brown Norway in the F1s, these animals will now live in excess of 2 years, and some of these animals we don't see any tumors at all. So it's dramatically different in the F1 compared to the congenic for Brown Norway. Whereas ACI seems to have, it gives us the most number of tumors at the earliest time point. And so a lot of our quick studies, especially studies that are looking at response to chemotherapeutics, and if we wanted to follow any molecular markers, we're using this F1 now to have a fully informative F1 that where all of the offspring are genetically identical, but we can still follow molecular markers throughout the genome. So now I just want to conclude here by talking about hormones and the sexually dimorphic phenotype. I pointed out in the genetic, in the previous slide, that there is this, seems to be a dramatic difference between males and females. And we wanted to know if this was specific to the rat or if this is something that is more ubiquitous. And at the time, a paper came out right before we were about to publish our results. It was an epidemiological paper studying humans and looking at a large population of people, and this was a generalized colonoscopy screening effort. So they screened thousands of individuals, and they found a very intriguing thing. Even though the reported number of deaths and cases of colorectal cancer seemed to be the same between men and women if you look on a large epidemiological scale, it was very curious to them that at any given age group, you had to screen nearly twice as many females to find the same number of adenocarcinomas and even more advanced adenomas at any given age group. So the total number of cancers identified in men and women were roughly the same, mostly because there are more women alive at any of these given age groups. So clearly there is some protection in women or a sensitivity in men that we wanted to see if this panned out with our rat model as well. There was another study that many people are, anyone who has studied breast cancer knows quite a bit about. This is the big Women's Health Initiative trial of the combination estrogen and progestin hormone replacement therapy postmenopausally. And the number that always comes up is this invasive breast cancer. There is an increased risk of invasive breast cancer with this combination therapy. But something that's often overlooked is the fact that in colorectal cancer, there's a very strongly protective effect, or it appears to be a very strongly protective effect in women that have undergone this combination hormone replacement therapy. So is this a hormonally protective effect? Well, if you look at the APC-Min mouse, we really didn't see a difference between males and females like we did with the Pirc rat. And so it was very curious to us. Now, this is total tumor number. This includes small intestine and colon, but we really didn't see any difference here. So we pursued this in the Pirc rat by doing a combination ovarectomy and hormone replacement. So we try and remove the hormones by ovariectomy or sham ovariectomy, and then replace with either, say, 17-beta-estradiol, medroxyprogesterone, or the combination of the two, or a placebo. We used 90-day release pellets. These are single replacement 90-day pellets, but we replaced them after 90 days, so we got a total of 180 days treatment. We did something similar in the rats. We didn't actually replace them with hormones, but we wanted to see if male hormones were potentially playing a role, so we gonadectomized rats and also non-surgically treated animals. So what did we see? So this is the standard Fisher 344 total tumor numbers for males when we were looking at the 210-day time point. So you can see that the number of tumors dramatically increases as you go on in time. So the longer the animals live, the more tumors they have. In females, it's dramatically less. But none of the effects, either the sham plus placebo, OVEX plus placebo, OVEX plus MPA, E2, or the combination, seemed to have no effect. So we ruled out the possibility that the female hormones, specifically estrogen or MPA, were having any effect on tumor initiation and actually tumor formation, really tumor growth. What was interesting is when we looked at the castrated male population, that number dramatically decreased and brought it in line with what we see in the females. So now we actually believe that testosterone is playing a potentially dramatic role in the formation of colon cancer, which was not previously recognized. And actually, this is now in preparation for publication. It's in the hands of my collaborator, Gijs van den Brink, and his graduate student, Jeroen Heijmans. And but when we wanted to know, is this really rat-specific, or is it also present in the mouse? And when we actually go back and look at now just colon tumor number in both the male and female animals, we actually see that females do get fewer colonic tumors than males. And this is anecdotal for a lot of people, because a lot of people look at the mice and maybe even only study the B6-Min male population and not females. But in fact, there is a statistical differences between male and female. They do show the same effect. But when we did the power calculation on it, you really have to look at five times more mice to see this very small difference between 0 and 1 or 1 and 2 average between these two groups. And for the same number of Pirc rats, we could actually get away with about 5 times less. So the power calculations that we did were based on really 10 animals in each group, and so 10 Pirc rats is enough to see the difference between male and female. So in summary, the advantages of the Pirc rat model, just to give you a summary of what I've talked about today, they have a mutation in APC, which is present in approximately 80% of sporadic colorectal tumors, as well as predisposing in FAP patients. These tumors arise primarily in the colon in the rats. We can use longitudinal analysis to study tumor growth by optical colonoscopy, including biopsies in which we can obtain material for expression arrays, even protein. Histological analysis, we've taken biopsies and fixed them, and actually you can do staining for beta-catenin and any other number of antibodies. We've also done some studies on using CT PET imaging. We've looked in more detail into the mechanism of tumor initiation in the Pirc rat, and the majority are through loss of heterozygosity through somatic recombination. But we actually feel that the more interesting class is this 10% that is epigenetically initiated. this is becoming recognized in human populations that there is a subset of tumors that undergo epigenetic silencing of various tumor suppressor genes. And this is something I'm going to be pursuing in the future, but is, I think, has a really broad interest for both the study of epigenetics in general and epigenetics in cancer. We've developed some congenic lines. The Fisher 344, which is really a co-isogenic line, we developed it on the Taconic background. We've kept it on the Taconic background. We back-crossed it back to Fisher to get rid of any other ENU, potential ENU mutations that were in the genome for at least 10 generations before we shipped it off to Taconic. So we feel it is a very pure co-isogenic strain that's available from Taconic, so there are no genetic background effects if you're studying just the Fisher background. But we have developed an ACI susceptible strain that really, the males only live to 180 days. They get more tumors and more aggressive tumors when treated with DSS. And then we have a brown Norway Pirc resistant congenic as well. As I said, some of these animals will live up to 2 years, especially the females. And in fact, we've seen some dramatic invasive tumors in these older females. There is a strong sexually dimorphic phenotype that is present also in humans. We feel that it's the same type of response, and so this is going to be the other aspect of what I'm going to be studying in the future. And I'd like to acknowledge really everybody. Of course, Dr. Dove for allowing me to do this work in his laboratory and really kind of shifting the focus not completely away from the mouse, because we certainly know that the mouse is useful for some experiments, especially looking at different genetic mutants that are present. But we're now doing a considerable amount of work in the rat. I have to thank Lawrence Kwong for helping with all of the initial identification of the Pirc rat and all of the initial phenotyping. Kathy Krentz here at the UW has been involved in a lot of aspects of embryo work and bringing rats from Japan for us. Linda Clipson for all her work in analyzing data. Amy Irving is a graduate student that did much of the vitamin D work in the rats. Elisa Weiss did the male gender work, or helped me with that as an undergraduate. Anthony Hunter and Corey and Brianna, all undergraduates here at the UW that helped with all aspects, or many aspects of the studies that we've done. And of course, I have to thank Michael Gould for really supplying the initial ENU mutagenized rats to us for screening. Michael Dutton for all of the statistical help. Tadao for some of the rat strains that we got from Japan, and of course Heis and Jerome for support with the gender studies. And of course I have to thank the American Cancer Society for helping to support my work. And if you didn't hear about it, in 2009 and 2010, the rat actually made it to the Jackson Labs for the techniques and modeling of human colon cancer. It was actually the first time rats had been in the laboratories, in the training laboratories for sure, but it had been a very long time since the Jackson Lab had seen rats in its thing. So we think it was very beneficial. This is me, and this is Amy, the graduate student who was helping in 2010. So it was a great group of people that came to Jackson Labs in 2010, and we think it was very beneficial for everybody involved. So with that, I think I will turn it back over to Megan and potentially get some questions.
Thanks, Jim. That was a really great talk. I definitely learned quite a bit. We do have time for a couple of questions. Please remember to go ahead and type your questions in the Q&A panel. We do have a few so far, but please don't be shy. Go ahead and type them in. Our first question, do the tumors metastasize?
Yeah, that's actually an interesting question. Now, because they have a mutation in APC and no other mutations, we really didn't have an expectation of metastasis unless there was a combination of multiple other mutations that would arise. And we didn't see any metastasis in the Fisher line, although we didn't look very closely. We didn't see any overt tumor metastasis. Now, in the brown Norway congenic line, we actually Amy just identified an older brown Norway female who was over 2 years of age just last week, and upon necropsy we found actually large tumors in the lung. So we actually feel that those may actually be the first metastasis that we see. And it may involve a very long latency, just like we see in the human, that long latency is really required for that transition to adenoma to adenocarcinoma. But that being said, and I didn't talk about all of the other resources now available in the rat. The rat has become a very strong genetic model. Later this week, there's going to be a conference at Cold Spring Harbor on rat genetics and genomics, and there's a large group at the Medical College of Wisconsin in Milwaukee that are developing either zinc finger targeted mutants. They've also developed ENU mutants and Sleeping Beauty mutants. All of these mutants they're making available to anyone who wants to study them. And what would be interesting to us is really the combination of those mutations with APC to see if we can promote progression. And we feel like now is the time that the rat is becoming much more adaptable to this to the genetic world. I mean, it's really become a very strong model now for these genetic tools that were really only available in the mouse before, but are now becoming available in the rat. So we can take advantage of both the larger size of the rat and some of the genetics.
I definitely agree. It really is an interesting time with the new technologies. Here's another question. So can you talk a little bit about why the tumor distributions between the mouse and rat are different?
Yeah, it's actually an interesting question, and we actually don't know. We don't know if it's a physiological difference between mouse and rat, or if it's something about how the tumors initiate. We really don't know. When we look at tumors in the small intestine in the rat, there are far fewer, and in fact, in the F1s that we've been looking at, if we see them, they're in the males, and there may be 4 or 5 tumors in the small intestine, and they're very easy to identify because they're much larger than what we see in the mouse. So to be totally honest, we really don't know. I mean, it's a wide open question, I think, and whether the mouse is just a different entity, either in its genome. We know that the chromosomes and how they're structured are very different than what we see in the human. So chromosomes and chromosome behavior in the mouse, chromosomes are acrocentric in the mouse, where the telomeres, the centromere is located near the end of the chromosome in all of the chromosomes. The rat is much different. The rat is a metacentric karyotype, much more similar to human, where the centromeres are in the middle of many of the chromosomes, and their behavior through these recombination processes may be very different in the rat compared to the mouse, just due to its structure. So it was another factor when we considered doing the rat, that it was a very different beast, both in its genetics and genomic makeup.
Okay. And in the Fisher version, how early do you see tumors?
Yeah, that's a good question. So I showed you on one Fisher where it looked like the tumors were very small, but by colonoscopy, we can actually see tumors at about 45 days of age. So very early lesions, and actually some, I mean, these are very obviously clearly adenomas. It's not just a small bump on the wall. So as early as 45 days, by 90 days, we do see a definite collection of adenomas in the colon in the males. Females, again, are a little bit delayed. So we can see them earlier. We're taking the Fisher 344 Pirc line now, crossing it to the ACI strain, and we're really terminating all of our studies at 6 months at the most. And so this allows us to do shorter time frame experiments, but really get the maximum use out of the rat.
Excellent. Thank you, Jim. I think we're running out of time, so we'll go ahead and end the webinar here. It was really fantastic. Thank you so much for presenting your results today. For the attendees, thank you for joining us. We will send out a link to view the webinar later in case you want to review it, or send it to friends who may not have been able to attend. So thank you, everyone, for attending today.